You are tuned in to Relativity.
Wait, are we live? We're live. Let's get real.
Relativity. Realitivity. Relativity. Good afternoon, Petra. It's been a while since we've done a Relativity podcast, and some good reasons. We wanted to take some time and develop our audio capability and our editing capability to be much better and provide you a much higher quality sounding and quality put together podcast. And we've done that. And so I just want to really thank Brittany Gallagher. She spent some time with us. She didn't have to. This was mentorship to us to get us set up, got us software, told us, a lot about how to do the production aspects of things, and was just lots and lots of great advice. So you'll see a much better and much improved audio to our podcast. So thank you, Brittany, for that. And I think with that, Petra, our next episode is
CRISPR. Yeah, huge thank you to Brittany, and hoping that we can do her proud with this one. So CRISPR, we have decided to split up into 3 parts because we think it deserves at least that level of detail and probably more. If you don't know, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is so catchy. I'm going to have to keep saying it.
say
The
it 5 times really
episode
fast.
that we're going to do here. Yeah, seriously. I can't believe I got it out that time, actually. The first one, we're going to cover some basic cellular biology, probably stuff you learned in high school and don't really remember, don't have a reason to use it every day, but it's super fascinating. We look into the genetics and how DNA, RNA, and basically the structure of your cells make you who you are. And then from there, we're going to look into the specifics on how CRISPR works, how it was developed, and it's a really fascinating story of all these different experts coming together and each chipping away a piece of the puzzle and making a really revolutionary technology. And then finally, for the third part, we're going to look into the future of CRISPR and how it's being used today, how it might be used and abused in the future, and all of the places that this technology could really take us. Did I miss anything there, Mitch? What do you think?
No, I think that's exactly the plan. And I find it so interesting that you brought up how this is such a great story. It really is. It's almost the story of CRISPR almost follows like a Michael Crichton type novel, right? That sort of filled with lots of technology and science, and then there's all this aspect of characters that have all come together to pull this technology together. And I also think it's interesting to call this a technology. It's literally, the way that a bacteria works, right? It's a bacteria's own defense against viruses. And so this just existed, they found it, and now the technology wrapped around that is so promising. So I'm really looking forward to all 3 episodes.
Me too. They're gonna be great. It's another cool instance of humans learning how to replicate what nature has done all along. And speaking of nature, let's talk cellular biology. Do you remember your high school biology class, Mitch? I barely do.
Not only do I remember my high school biology, I happened to be a biochemist and took plenty of biology in at the university, right? So I had cell biology as one of my courses. I had genetics and a handful of other things that will be very appropriate for this. And I won't say that I remember it all, but in preparation for this, it brought back lots of good memories of going to school and lots of memories of how all this stuff works. And it got me really thinking. This was a very interesting topic. So look forward to discussing.
Well, I'll just say that I'm grateful to have a biochemist in the room because it's been a while since I got to think about cell structure and how our DNA gets replicated and all these things are absolutely fascinating though. So I guess if we start at the top level of a cell, the highest level that we can start at, you want to jump into what the nucleus is?
I'm gonna take a step back from that, Petra, and really just talk about the cell in general. Maybe hit the nucleus right after that. I think it's really important for people to understand that we are made up of trillions of cells. That's the basic building blocks of what we are, right? And so to get there, you need to envision the cell kind of in 2 parts. You've got the cell itself, kind of this amorphic blob, if you will, and then inside of that is the nucleus, and that really, in my mind, is equated to if you just look at an egg, right? If you're going to fry up an egg, the yellow yolk is the nucleus, and then the outer white section is the rest of the cell that contains all the various organelles and everything that we have that really are producing all the proteins and the enzymes and everything else that is making up our muscles, whether it's a hair follicle or blood cells or whatever the case may be. So if you can keep that kind of image of a egg in the frying pan, if you will, that's a great kind of analogy for the makeup of a cell here for us. The nucleus is definitely one of the more interesting parts because that is where our DNA is located. And do you remember, Petra? I mean, I remember back in high school just pulling cheek cells, right? We would take a little swab and pull cheek cells, and you could actually see the DNA in those cells.
Yeah, we actually did an experiment. It all came back to me a little bit as I was reading through this and in preparation. Not totally, but we did a similar thing to isolate the DNA of strawberries, which I thought was really cool. And
Oh,
I
interesting.
kept the resultant kind of vial. They let us take home the vial with the DNA strands in there, and I kind of kept it for a long time because it's just this powerful reminder of how magical and elegant nature really is, that a being as complex as a human, or as complex in a different way, I guess, as a strawberry, can be encoded in strands of DNA. It just kind of hit home for me.
All right, so that's pretty interesting that you checked out a strawberry. I didn't even think about looking into something like that. So now let's talk about DNA itself. And the way I kind of look at DNA is the blueprints, if you will. It has everything encoded in those strands that you can see underneath the microscope, believe it or not, that makes up every aspect of who we are. Not only does it make up all the different aspects of like this is a muscle and this is a blood cell and everything else, but each one of those has certain qualities that give us green eyes or brown eyes or blonde hair or black hair or whatever the case may be. It's absolutely amazing that this little strand of molecules can actually code everything about a person, about a plant, about a bacteria, or about a virus. It's absolutely amazing.
It really is. And if we think about the structure of DNA, I think most people have probably seen that double helix where there's the almost a ladder shape that's twisted. And it's fascinating to think that in all of the complexity of humans and the world, there's only those four pairs of bases that form the ladder rungs of a DNA strand. Actually, I remember this fun thing that my high school biology teacher said, which was that if we were ever in a body snatcher situation where we had aliens that were impersonating people, I know it's far out, but if we were in that situation, you could tell if an organism was from Earth because if they had more or less than four types of bases in their DNA, then they wouldn't be from Earth. Every being, every living thing on Earth has four types of bases.
Yeah, and I look at that basis sort of as it's almost like the binary language of a computer, right? Where the sequences of the ACTG combinations are really what, when we go through and read that, that's really telling you what that protein is going to be, or however this is going to be expressed. So I guess we should do a quick definition here: a gene and an allele. So a gene is something that is specific to something like your eye color. And the allele is actually the expression of your eye, of what your pupil eye color is going to be, right? So this was a study that was done a long time ago by Mendeleev, right? Where I think most of us were familiar with this in our high school studies or in the college studies is, he wanted to mix these plants. And he thought that if he mixed a white flower with a red flower on these pea plants, that he would get a pink flower. And what he saw was is the exact opposite. He still got red and white. And what he came to find out is that there are different sets of alleles, and they can be either dominant or recessive in the way that they are expressed. And this is very, very important to understand as we go through this, because we are all inheriting 50% of our genetic makeup from your father and 50% from your mother, and how those come together and what is dominant or what is recessive is what gets expressed.
Right. And I studied the history of science. I'm no biochemist, but I live for these stories about the people who kind of created the technologies or the fields that really define our world today. Gregor Mendel is such a fascinating fascinating instance of this because he was a monk, right? And he was experimenting with pea plants just to grow different kinds of food and figure out better ways to farm where he was. And he really did not reach any kind of level of fame while he was alive. And yet, think of how fundamentally he's transformed our understanding of the world. And it's just a fascinating case, actually, like much more of CRISPR that we'll talk about in the next part of this 3-part series here. But it was really curiosity driven science, right? It wasn't science in pursuit of profit. It was science in search of truth and in search of answers. Anyway, I just think it's such a fascinating story.
Absolutely, and actually the stories that behind all of these discoveries were very interesting, and I think we should talk a little bit about Watson and Crick, very famous, if you will, for finding the structure of DNA. But there was a big story behind all of that too. Did you look into some of that and know some of that history on that story as well?
I'd love to hear what you found on it.
I think one of the things that they get a lot of the credit, right, for this, but it was actually a much more involved, and many more players really played into the discovery. And I think it was so dang important that we figured out what the structure of DNA was. And I think we'll see that kind of thread throughout our discussions, as there's a lot that goes on with geometry in chemistry and how you interact, how things come together and work together. So a lot of this came through with the X-ray crystallography that was taking place. And dang it, for the life of me, I had her in my notes right here, and I'm already forgetting. Do you remember the
Rosalind Franklin.
Rosalind Franklin? And her work was instrumental to
Yes.
this. It was, it gets downplayed quite a bit, or it doesn't get the accolades that it deserves. But the ability to actually visualize this double helix was so dang important to understanding how it unlocked the mysteries of DNA in so many ways. This was a great story of how this all came together. I kind of always think of these great mysteries and this how they are solved as taking years and years and years. But the reality is once they kind of got onto the scent, if you will, of this discovery, things started happening pretty fast. And they went from, okay, we're going to figure this out, to it was figured out in a very short period of time with the help of many folks in there. So very interesting time.
I think it's fascinating though, because there's a lot of stories like this. And it's part of the reason that we do this podcast, right, Mitch, is because we tend to think of history as these unique, isolated events. Watson and Crick discovered the double helix, but the truth is always so much more complex, and it always involves more people who are making equally important discoveries to pave the way for a discovery like that, right? It's never just one moment in a vacuum. It's this whole story of human progress, and I think it's fascinating to kind of go into those side stories that really define what Watson and Crick and other scientists were able to do.
Well, and all of this is just so complex. There's no way that one single individual could ever have all the knowledge across all the different fields to make this work. Like the crystallography, the X-ray crystallography techniques, that's not a biolog that's not some, a biologist isn't going to be necessarily the person you would think of as the experts in that field. That's straight physics, right? So you've got to have a deep background in physics and that kind of understanding of how all of that works to pull that in. You got to have the biochemistry, you got to have genetics and whatever form of genetics that they had in the 1950s to pull this together. The math, it's highly complex, to the point
It
of
really is.
they had the machine shop on standby as they were building. Did you get to see those pictures? I'm sure you did, of Watson and Crick standing next to their model. They had the machine shop there at the university, just kind of on call, and like, okay, guys, this is what we're thinking. This is how we want to build this up. And they were building it as they went. It's such a great story.
It really is. We might have to devote its own episode to
It would be interesting. Yeah.
to DNA.
Yeah. So I think one of the things, so we talked about basic genes, and then the alleles and how all that's expressed. We should probably dive in here to talking about what goes on. Let's actually talk about a couple of things here. Let's talk about what are germ cells and what are somatic cells, because I think that's an important distinction to understand, especially when we start talking about the ethics of this later in episode 3. Petra, do you know the difference between those two?
Yes, if I remember correctly, and I didn't double check this, so this is like a fun pop quiz on
Yeah.
the air. A
Here's a
germ
game.
cell is basically involved in sexual reproduction, right? So it gives
Yep.
rise to the gametes. Is that, and then the somatic is everything else,
Yes, that's correct. So the somatic cells
everything
are everything
except.
else.
Oh,
Yeah.
woo!
Perfect. Yeah. And so things that we're going to be talking about here obviously is gene editing. That's what CRISPR is all about, but there's many different ways of doing gene editing as we'll talk about. But when you make a change to a somatic cell, that's not a change that's going to be hereditable. But if you
Right.
are to change something in a germ cell, that is something that you will pass on. So that I think that's something very important to remember as we start getting into this deeper. I think one of the things also to understand is that every cell is actually capable of being any other type of cell. You could have from heart tissue to a liver to a blood or whatever the case may be. Early in our development, what's known as the stem cells, they basically program what all the others are going to do. So the information is there for anything to be whatever type of cell it is, but there is some fascinating mechanism, and who knows what the heck that is, that actually says, nope, you're going to go be a hair, and you're going to go over there and be, in the skin, and you're going to be blood, and you're going to be liver, whatever the case may be. That mechanism is, well, it's mind-boggling if you ask me. It's pretty
Yeah.
impressive that we have this type of, the cell itself is just a fascinating machine. And the idea that that we have this ability that these cells, these little tiny things with no brain, have the capability to go off and be whatever they need to be to make a plant or a human or a dog for that matter. So that's pretty dang
They
interesting.
have no brain, but they make up our brain, which is
That's
cool. um, yeah. It was cool. I was reading about genes, which I think we already defined, but basically it's the DNA sequence that contains instructions to make a protein, right? So the size of a gene can vary greatly. Sometimes I
Yeah.
believe they can range from 1,000 bases in that DNA double helix to 1 million bases in humans at least. But genes themselves only make up about 1% of the DNA sequence, which I didn't think I knew before. And then the DNA sequences outside this 1% are what's regulating when and how much of a protein is made. So right, that's that 99% of DNA is telling the cells how to use the genes, the 1% of it, which is just fascinating because we always fixate on that identity piece, right? The gene piece, but really, as you said at the beginning, any of these cells can become anything, and it's really up to the DNA to determine which cell becomes what.
Well, and I think another fascinating part of this as we learn more and more is that these genes need other genes to be expressed in certain ways, right? So we're just barely getting an understanding now of how the genes are interacting amongst other genes and how all of that is being turned on or off depending on what's present. Just recently, probably a couple weeks ago, actually probably more like a month ago, right? They announced that the Human Genome Project is now complete. We've got the full map of the human genome, and when you start looking at that, we've learned so much. I think the Human Genome Project started about the time I started some of my genetics classes and everything at the university. And so, it's taken quite some time. It's well over 20 years to map everything out and be confident that we know what all the different genes and stuff are. But now that we've done that, we've got even more mysteries to solve. And how do all of these interact and work together? Because there's definitely more coding out there than.
Yeah.
Okay, so I think, Petra, we need to jump into talking about how exactly do we make copies, and how exactly do we make proteins and the other things based on DNA and RNA, and specifically what are the roles in all of this?
Yeah, absolutely. So, one quick piece that I think helps to understand this are thinking about organelles, which are that subcellular structure that has multiple specific jobs that it usually performs in the cell. So you can think. I'm pretty sure this is accurate, Mitch, but you could think of an organelle like an organ in the body, right? It has various roles, but they're specific. And then like for example, a nucleus is a cell organelle which stores the genetic information, the DNA. The mitochondria are the powerhouses, right, of the cell,
Yeah, the
which produce
powerhouse.
chemical energy. Yeah. And then
producing that
the ribosomes
ATP.
are the ATP. There you go, biochemist. And the ribosomes are the part that's actually assembling the proteins that are encoded in DNA and transcribed by RNA.
Yeah, and I think not necessarily an organelle, but another thing to really understand is that the cells wall on the outer cell as well as the nucleus are both permeable, and they have a whole system of gates, basically little chemical gates that allow things both in and out. And so that's how we can move the bits and pieces that are needed to make a protein into the cell and then transport that protein back out to where it needs to go, and make an eyeball or make a hair follicle or whatever the case may be a part of the liver. And that's also how we can move, RNA into the nucleus so that it can pick up the information it needs to go back out and work with the ribosomes and start making the proteins and other things that we have out there. So I think that's an interesting feature to.
Absolutely. It is interesting. And I guess while we're talking our way through this here, I want to make sure that we're defining like what a protein is, which actually
Sure.
is quite interesting, right? So they're large molecules or complex molecules, I should say, and they do most of the work in our bodies, right? And they're made up of amino acids, which are the building blocks of proteins. And I believe there's 20 types of amino acids. Mitch, is that right?
Is it 19 or 20? It's one of those, but it's, yeah.
It's one of those.
We will put it. We will
And the
get the
amino
exact number in the
acids.
show notes. I used to have that memorized. For whatever reason, we had a professor that was all into memorization, and for the life of me, I can't remember the number right. But they're pretty important.
All right, well, we can double check.
I think one of the interesting things, though, too, to kind of discuss right now, and it's a little bit of a rabbit hole, but we like rabbit holes. I, I think when you start understanding that, that the amino acids are what's making up the proteins that are basically making up every part of our body, it's so important to understand that you have to eat the foods. Your diet has to consist of the things that actually are building up your body. So if you have a very unhealthy diet, if you're not drinking water and you're not getting all these amino acids and the other things that are needed to do the cellular cycle, just think of how that will affect you overall. Right? You have to give these machines the good stuff to make the body work the way it should. So I just want I think that's very important for other discussions that I'm sure we're going to have at some point in time about nutrition and how the body works in general. But I wanted to put that in here because it seems like the right place as we talk about how we build in all the different parts of our body.
So funny enough, fun fact: the white potato contains all the essential amino acids that you need to build proteins and repair cells and fight disease. But I'm not saying that you should just eat only white potatoes because you would run into other vitamin and mineral deficiencies. But isn't that cool that they actually have all of them?
Yeah, that is pretty cool, and that's another good point that you made. There's a lot of minerals and stuff that are also needed. There's all these calcium channels, potassium channels, and all these other things that are made up of these minerals that actually help us regulate not only neurons firing. We need that the electricity, if you will, to make the neurons work. The differences between a plus and a minus, the ions on some of these chemicals, as well as the gates I was talking about that let you in and out of the cells. Get your vitamins and minerals as well.
Nice. That was awesome. So let's talk about DNA and RNA. So we talked about DNA, right? That double helix. So I guess structurally, Mitch, what's the difference with RNA?
So it's basically they're both the two sugars, right? But the DNAs we're talking about is in that double helix, and the RNA is just a single strand. And I think that
Right.
that's one of the most important parts of all of this is in the way that they can come together. And so we've got several different things going on. We need to make new cells, and so when we make a new cell, we're basically making an exact copy. When we do that, we actually have to make copies of the DNA. It's a really fascinating process where we go in and they actually unzip the DNA. And if you think about this, when we have our pairs, right, the AC and the TG, those pairs are basically making a mirrored copy. But you have to remember if I went from the top of one to the bottom, and then if I went from the bottom of one to the top of one, you're going to have the same information in that code, right? Because of the pairing that they have. And so you're going to see that there's a 5' and a 3' on your strands of DNA, and that tells which direction that the enzymes and the RNA and everything else are going to go in and read that DNA. So what we see when we want to do a cell division, right, is we open that up and we split. Then we have this group of contractors come in, if you will. They start making the new strands of DNA so that when they come back together, now we've got basically two new sets of DNA, or four total strands that have come back together. Then the cell can go ahead and divide, and you have all the same genetic information sort of saved, if you will, from one cell to the next.
Right. It's so cool and elegant how this works. So I love the visual of the helicase unzipping that DNA double helix, and then the RNA copying one strand. And because we have those four different nucleotides, then and each pair forms a complementary pair with the other pair, um,
Yeah,
then it's automatically replicates the same structure. So the mnemonic device that my high school biology teacher used was at the golf course. So A and T
there you go.
will always pair with each other, and G and C will always pair with each other. It's gosh, can I remember their names? Adenine, thymine, guanine, cytosine is the golf
Got
course.
him. You
Look
got
at
him.
that! I haven't forgotten it all.
Yeah, and so some very interesting things too that we have going on here is we have all kinds of different enzymes coming back in and actually looking for errors in the transcription, right? So it's going back and forth, reading these codes and making sure that we have good replication. And this is one of the things that this is our body trying to make sure that we don't have bad mutations or things that will cause, cancerous cells. And all cancer really is just the kind of division of cells running away with itself, right? It's lost its ability to fix itself, and it's just making a mass of these cells that don't. So these massive cells just have some sort of issue with them that they're no longer doing what they were supposed to, and they're just replicating at a very high rate. And it's amazing that there's this
Right.
enzyme that can go in and do that type of stuff.
Right. So I guess I'm pretty sure that enzyme is kind of a catalyst, right? That our
Yes.
bodies make to produce a specific reaction in the cell. Is that a reasonable way to define it?
Absolutely that. And so they're responsible, and what's really interesting about the enzymes and proteins and stuff in general is that they all fold once they're made. They all fold in these elaborate, and there's great illustrations of this on the web, but they will fold in very unique ways, and a lot of times by the way that they fold and the way that they were encoded, they will do very specific work either at the during this whole process of reading our DNA, whether or not it's reading it to do the cellular division, or whether it's reading it to make more proteins or whatever else that the body needs. And so these folds, they allow these certain chemical interactions to happen, and they kind of just wrap themselves around the strands of DNA and just zip right down them and do the job that they were set up to do. So folding is another topic within biology of proteins and everything that is getting a lot of interest and work, especially now that we have such great computer processing capability. It's much easier to go in, or it's much faster, I guess is probably the term, to go in and really start considering how the folding and the chemical interactions are taking place to do the jobs that they were meant to.
So interesting on protein folding, and we could totally go down this rabbit hole. But Google actually just open sourced its protein folding software. They had built a software system that would try to figure out how proteins are folded. Because as you said, it has serious implications for how those proteins express themselves, which is related to disease and all other kinds of things. But it was DeepMind at Google that Google had acquired, and they began a system called AlphaFold, which figures out what kind of shapes the proteins are folded into. Anyway, it's been a huge challenge in biology for decades, and it's really fascinating to see Google apply their brains to this problem.
It's going to be huge steps in the knowledge and capabilities and just the new technologies. It's going to take us all new ways of solving our problems and how we fight disease and everything else. It's so interesting. I look forward to actually learning much more about that. You'll have to fill me in, or I will
For
have to do
sure,
some
for sure.
research. That sounds like a very interesting topic.
Yeah, both. But back to the cell. So let's think about the other things that will help us understand CRISPR, right? So I think I mentioned I think I mentioned the helicase earlier, which is an enzyme that unzips the DNA double helix so it can be replicated. So it turns it from kind of a zipped zipper into an unzipped zipper, right? And then another enzyme that's important is a nuclease, which is an enzyme that can actually split or cut the chains of nucleotides that make up DNA and RNA, and a nuclease can cut them into smaller units.
Another point, Petra, that's pretty interesting here with all of this is that DNA is fairly consistent. Like we talked about, it's always trying to, we have tools in the cell to make sure that it stays the same from time to time. But there is some really interesting things, and this is how we're actually able to develop our antibodies in the lymphocytes, right? And this is our in the blood cells. And this is an area where DNA can actually have some variations and it changes over times. And this is how our antibodies are actually working to, attack infections of whatever source that they might be. We can have and we can see serious or significant rearrangements in those cells in our immune system.
Right. So thinking about the immune system, we have an innate immune system, right, which is basically what we're born with to help us fend off disease. And those we have all these different kinds of defense mechanisms in our cells, but we also have an adaptive immune system, and that's where our cells see new viruses or new things that we need to be defended against, and it actually will take a snippet of that attacker's DNA and basically encode it into its own DNA so that if that attacker ever returns, then the cell will know how to fight it. That's a very simplified version of what happens.
It's almost like caller ID, right? It's we'll recognize it when it happens
Yes,
again, and we'll know whether we're going to answer that phone call or not. So, I think one
I
of the
love
things
that.
too, we've got a good line going here, but I think we need to kind of step back too and talk a little bit about the difference between a prokaryote and a eukaryote. And I think that's going to be important as we move forward into part two of this discussion. We are eukaryotes, right? We are an organism that is made up of many different cells. All these cells look a little bit different as they go from, whether it's your liver cell or what have you. So the differences in prokaryotics is that these are singular cellular organisms. This is a bacteria as an example.
Absolutely. And in prokaryotes, they're generally smaller than eukaryotic cells, but the genetic material isn't stored within a nucleus that's surrounded by a membrane, right? So I think it's basically stored in a nucleoid that floats in the cytoplasm of a cell rather than being kind of wrapped in this membrane as our eukaryotic cells are.
And I think one of the things to talk about there too is they replicate at very high speeds, right? And you can see everything that's going on in a prokaryote in a very short period of time because they're just replicating, and that is the lifespan is short, but also the time in between each replication. And so this is why a lot of scientists do tons of study work in genetics and a lot of different fields in the bacterias, right? Because you can grow, a whole host of bacterias on just a plate of agar, and overnight you're going to have millions of cells, if you will, of the prokaryotic cells to do your studies on. And so you can quickly see how changing, whether it's gene editing, genetic editing of some sort, or whatever else that you're trying to do, how it reacts to a new environment or whatever the case may be, whatever you're studying, you can do this very quickly. It's sort of like we use mice to study, eukaryotic organisms, ourselves or whatever, to see how pharmaceutical companies will use that to test their medications, et cetera, because the lifespan of a mouse is fairly quick. So it's the same concept using these prokaryotes in this science.
Yes, and they're quicker once we experiment on them. Their lifespans. I
Yeah.
actually have been reading some cool research on what's another diversion here, but on what's called in silico testing, and that's basically using computers
Yeah.
to simulate human reaction to drugs instead of using animals, because obviously animals are, well, they're what we've always used, but it turns out that as our computing systems get better and better, we might actually be able to replicate how a human would respond with more accuracy than a mouse can tell us what the likely response will be from the human body to a new drug. Anyway, super interesting field that I'm
No,
following
I
along
actually
with
think
as
in
well.
silica's, yeah, I think in silica is going to be phenomenal. First off, I just like the name in silica, sounds pretty cool. But if you think about that,
It's
some of these
awesome.
things that we want to do, you're not able to see. So in gene editing, we're not able to see the effects of this for tens of years at times, right? So if we're trying to work on something for a dementia or Alzheimer or something like that, how in the world can we do that test, right? We got to wait till what, somebody's 60, 70 years old before Alzheimer's starts to show up. You That's no good. So I think things like in silica is going to be tremendously important. I also just think that this basic understanding of our genome, as we know more and as we can model things and as we can start to understand sort of these codependencies or interdependencies between the different genes and how they're expressed, and we're seeing lots of examples of where if this gene is present in the presence of this other gene, then this will turn on, or maybe you're not as susceptible to a certain disease or a certain illness, a certain genetic disorder. And I think being able to play this out on the computers is really, really going to change our understanding of our bodies, of the just life in general. And we're really going to have some great capabilities to help us live a better life and prevent some of these diseases and other things that just don't make life that great.
Interesting to think about how we're basically learning nature's secrets and how genetic material works and how it's copied and how these enzymes kind of can prevent viruses and attackers from being copied. And then what we're using is our own human intuition and intelligence to basically supercharge what nature is teaching us, right? Because we're saying, oh, look, this is how nature does it. So can we speed up nature in a way that improves our lives and improves the lives of those who will come after us? And of course, there's a lot of ethical questions that I can't wait to get into in the third part of this, Mitch, where we're thinking about what each person considers to be a good development might be different than what someone in a different country or state or even the house next door would think is a worthwhile use of our time and energy and investment in this kind of technology.
Yeah, I'm actually really looking forward to episode number 3. There's a lot of stuff to discuss in that area. I think one of the other things that's so interesting too, as we start to understand, our genetics, it's going to lead to things that can really help us. I'm going to call it. I know a lot of people are working on this idea of living longer, adding years. I'm actually looking at it more in the for us. Maybe you being younger, you might look at it as adding years. For me, I'm looking at it as adding quality to my later years, right? So if we can understand
Yes,
these mechanisms, how cells deteriorate, how we can keep them healthier longer through stuff like in silica and through stuff of understanding the genome much better, and maybe doing some experiments, with the mice, but also combination with the in silica stuff. I would love to have a healthier brain, healthier muscles and tissue, at the age of 90. And just so that I can have a better quality of life, not just me, just all of us can have a better quality of life at 90. I think, there's also the potential of extending our lives. There's no reason. You look at the biggest mammals in the world, and whales, and they can live hundreds of years, turtles, hundreds of years. So there's no reason why we can't. And I'm sure that that's where someplace where science is going to go. And that might be something for your generation or maybe ones that follow will be going. But this basic understanding of cellular biology is really going to make life, it's going to improve so many people's lives in the future.
absolutely. And I guess that's a good step into our episode 2, where we'll talk about how all these different people came together to actually turn CRISPR from kind of a secret of bacteria, a secret of cell defense, into really an opportunity to transform our futures on the planet. And I totally could not agree more with your thinking about health span instead of lifespan, right? Like what's a long life if it can't be a good life? So hopefully CRISPR will help us on both those fronts.
Absolutely. So I look forward to episode 2. Like you say, this it's such a fascinating story, and the personalities are just so all over the place. Till the next one, Petra.
There we go, Mitch. Can't wait for it. Hey, this is Petra. If you like this podcast, please subscribe wherever you get yours. Also, let us know what you're interested in, and we'd love to dig deeper and explore topics that you want to hear about. Thank you for listening to Relativity, and tell a friend.